Welcome to Chapter 1, The Invisible Living World. Around us exists a fascinating world that cannot be seen with our naked eyes. In this lesson, we will learn what microorganisms are, classify different types, observe them through simple activities, compare their characteristics. After rainfall, the surroundings become rich in moisture. Such environments provide excellent conditions for many microorganisms to grow and multiply. Students collect water samples from ponds and puddles. Although the water appears clean, it contains countless tiny organisms invisible to our eyes. Using a microscope, we can observe the invisible living world. A single drop of water may contain hundreds of different microorganisms.
Have you ever wondered what would happen if the invisible world suddenly became visible? Tiny living organisms are present everywhere.
These green organisms are algae. They prepare their own food through photosynthesis and release oxygen into the environment.
These are fungi. They grow on moist organic materials and obtain food by absorbing nutrients from dead and decaying matter.
Protozoa are single-celled organisms. They move using cilia, flagella, or pseudopodia, and feed on tiny particles in water.
Bacteria are among the smallest living organisms. They are found almost everywhere and can be both useful and harmful. Viruses are even smaller than bacteria. They can reproduce only inside living cells, making them different from other microorganisms. Although invisible to our eyes, microorganisms play an important role in nature and in our daily lives. Let us begin our exciting journey into the fascinating invisible living world. Our eyes can see only objects that are larger than a certain size. Many tiny living organisms are so small that they cannot be seen with the naked eye. Good morning, students! Look around you. Trees, flowers, birds, and insects. But did you know that millions of tiny living organisms are so around us? Our eyes can see only objects above a certain size. Tiny organisms are much smaller than an ant or even a grain of sand. That is why we cannot see them without special instruments. First, scientists used a magnifying glass. It makes small objects appear larger. Look carefully at the ant. Can you now see its legs, antenna, and body more clearly? Observe carefully. The letters look bigger through the water-filled flask. This happens because the flask acts like a magnifying lens. This simple activity helps us understand how reading glasses and convex lenses help us see objects more clearly. Scientists improved these lenses over many years. Their discoveries finally led to one of the greatest inventions, the microscope. This is a biology laboratory. Here we use scientific equipment to study tiny organism microscope. It helps us observe organisms that are invisible to our naked eyes. This is the eyepiece through which we look. These are the objective lenses that the specimen. The stage holds the slide. The adjustment knobs help us focus clearly. Look through the microscope. Amazing! Tiny living organisms are moving in the water. These microorganisms are living around us every day, even though we cannot see them with our eyes. For thousands of years, people did not know that millions of tiny living organisms existed around them. These tiny things remained hidden because our eyes cannot see them. Long ago, people made an exciting discovery. They found that a curved piece of glass could make tiny objects appear much larger. This simple discovery changed the way humans observe the world. This curved glass is called a lens. When we look through it, small objects appear bigger and clearer. The discovery of the lens became the foundation for the invention of powerful microscopes. Students, today we are going back nearly 360 years. We will discover how scientists first entered the invisible world. Their discoveries changed biology forever. Today we about one of the most important instruments in biology, the compound microscope. It helps us observe tiny organisms and cells that cannot be seen with our naked eyes.
In 1665, an English scientist named Robert Hooke carefully studied tiny objects using one of the earliest compound microscopes. One day, Robert Hooke placed a very thin slice of cork under his microscope and carefully observed it. He observed many tiny box-like empty compartments. They looked similar to the compartments of a honeycomb. Robert Hooke called each tiny compartment a cell. This was the first time the word cell was used in science.
Long ago, people discovered that a curved piece of glass could make small objects appear bigger. Because it looked like a lentil seed, it was called a lens.
Exactly, it magnifies the text.
Over time, lenses became more powerful. Simple magnifying glasses gradually developed into microscopes, helping humans observe the tiny world.
Robert Hooke's book Micrographia contained detailed drawings of tiny objects that people had never seen before. It became one of the most important books in the history of biology. Around the same time, Antony van Leeuwenhoek from the Netherlands made better lenses and developed more powerful microscopes.
Using his improved microscope, Leeuwenhoek became the first scientist to clearly observe living microorganisms, such as bacteria and protozoa.
Because of his remarkable discoveries, Antony van Leeuwenhoek is known as the father of microbiology.
Leeuwenhoek also observed red blood cells, sperm cells, and the flow of blood through tiny blood vessels. His discoveries transformed our understanding of the microscopic world.
All living organisms, whether plants, animals, or humans, are made up of tiny units called cells. The cell is the basic structural and functional unit of life.
Today, let us study a plant cell by preparing a temporary mount of an onion peel. We will observe the onion cells under a microscope. First, wash the onion bulb, cut it vertically into 2 halves. Using forceps, gently peel off the thin transparent epidermal layer from the inner surface.
Place the thin transparent onion epidermal peel in a petri dish. Add 1 or 2 drops of safranin stain and leave it for about 30 seconds.
Now transfer the stained, thin, transparent onion peel into clean water. This removes the extra safranin stain and makes the cells appear clear under the microscope. Place the thin, transparent onion peel flat on a clean glass slide. Add 1 drop of glycerin to keep the cells moist and improve visibility. Place 1 drop of glycerin on the onion peel after that. Carefully lower the small cover slip at a 45-degree angle so that it covers only the specimen and prevents air bubbles. After placing the cover slip, gently remove the excess glycerin around its edges using blotting paper. This keeps the slide clean and improves the clarity of the specimen. Place the glass slide on the microscope stage and secure it with the stage clips. First use the coarse adjustment knob, then the fine adjustment knob until the onion cells become clearly visible. Observe the onion peel under the compound microscope. The cells are long, rectangular, and closely packed, giving them the appearance of bricks in a wall. Each cell
Notice how the onion cells are arranged closely together, just like the bricks in a wall. This close arrangement gives strength and support.
Just as bricks are the basic units of a building, cells are the basic units of all living organisms. The cell is called the fundamental structural and functional unit of life.
Today, we observed onion cells under the microscope and learned that all living organisms are made up of cells. The cell is the fundamental structural and functional unit of life,